Hello everyone. In this module, we will talk about the DNA structure. So this is the reference book that I've used throughout this presentation. You can go back to this book and review if you want to. Uh, but most of the image that I've used is from this book, and I found this book to be very helpful. So I would recommend to go back and review this book. Okay, so let's begin talking our discussion on DNA structure. So first of all, let me ask you a question. What is a DNA? DNA is nothing but it's just it contains our genetic material for our cells, and from DNA we can make RNA, and from RNA we can make the protein. So this protein is a constituent of the cell, as well as some of the protein act as an hormone. So this DNA contains all of the genetic material to survive. DNA is made up of a three structure. The first is a sugar, nitrogenous base, and the phosphate group. So on the right side of the image, I depicted the image here, and as you can see here, this is the pentose sugar here, and this is the phosphate. It is attached to a sugar, and then there is a nitrogenous base. It is also attached to a sugar. So the phosphate and the nitrogenous base is attached to a sugar. And one thing that I like to mention here is that the sugar that is present here in the DNA, it is the deoxyribose sugar, whereas in the RNA the sugar is a ribose sugar. Now, what's the difference between the deoxyribose sugar and the ribose sugar? So if you see the name, it's called the deoxyribose. So deoxy means the absence of oxygen. So in at the second position here on the pentose sugar, there is an absence of an OH group, as you can see here on the right side of the image. Whereas if the structure were to RNA, the OH group should be present here. So in the ribose sugar there is a two OH group, whereas the deoxyribose sugar there is an only one OH group at 3' position. So there is an absence of OH group at 2' position. That's the main difference between ribose sugar versus deoxyribose sugar. And nitrogenous base can be different in RNA and DNA. Although they are relatively same, there is only one base difference that we will talk about later in this lecture. And one thing also I would like to mention here is that these nitrogenous bases that you can see here, adenine, thymine, guanine, and cytosine, they are paired with each other via hydrogen bond, whereas the nucleotide, as you can see here, the whole nucleotide, which is okay. Before we move on to the nucleotide, let me briefly mention what is the nucleotide. What's the difference between nucleotide versus nucleoside? Okay, so nucleotide is phosphate, sugar, and the nitrogenous base. These three structure makes the nucleotide. Okay, whereas nucleoside is nothing but just the sugar and the nitrogenous base. So nucleoside sides do not have a phosphate group, whereas nucleotide have a phosphate group attached to a sugar and a nitrogenous base. Okay, so that's the difference. So now let me mention about the phosphodiesterase bond. Okay, phosphodiester bonds is a bond between this nucleotide, as you can see here on the right side of the image. So this is the sugar, this is the nitrogenous base, and this is the phosphate. So this phosphodiester bond connects this nucleotide to each other via this phosphodiester bond. Okay, so this is the take home, that the nucleotides are connected via phosphodiester bond, whereas the nitrogenous bases, they're connected via hydrogen bonds. And we will talk more about this hydrogen bonds in later in this session. Okay, and this phosphodiester bond, it connects 5' end of the nucleotide to 3' end of nucleotide, because as you can see here, let me bring my pen. Okay, so as you can see, this is 1 1, this is 2, this is 3, this is 4, this is 5. So this 5' end of this sugar here is attached to a phosphate, and this is this 1, this is 2, and this is 3. So as you can see here, this phosphodiester bond, connects the 5 prime end of a ribose sugar to a 3 prime end of another deoxy ribose sugar. Okay, I forgot to as I mentioned ribose sugar, but it's a deoxy ribose, and sorry for that. So 5 prime end of a deoxyribose sugar to a 3 prime end of the deoxyribose sugar. So this is the site where the phosphodiester bonds are connected to each other. Okay, so let's move on to the next one. Again, this is something that I have already mentioned. In case of DNA, the sugar is deoxyribose sugar, and in case of RNA, the sugar is a ribose, and I've already mentioned the difference between the deoxyribose sugar and the ribose sugar. Also, phosphate is attached to a 5 prime end of the DNA, and as you can see here, already I mentioned, this is the first carbon, this is the second carbon, this is the third, this is fourth, and this is the fifth carbon. And as you can see, at the fifth carbon position there is an attachment of the phosphate group. Okay, so this is something that is not really high yield, but it's very important to know. It allows you to differentiate the structures of the DNA. Okay, and as I said, there are four different type of bases present, and we have adenine, thymine, guanine. It's actually guanine, not the guanosine, it's a guanine, and cytosine. So in case of RNA, there is an absence of base thymine. Instead of thymine, there is a uracil present. So in case of RNA, adenine is base pairing with uracil, and guanine is base pairing with cytosine. Okay, and in case of DNA, adenine is paired with thymine, and guanine is paired with cytosine. And again, we will talk about later about this how this base pairing occurs via hydrogen bonds in later in this lecture. Okay, but for now, just know that this DNA contains deoxyribose sugar, and RNA contains sugar, which is just ribose sugar. Also, one thing that I want to mention is that this DNA is a double helix structure. Okay, as you can see here, this is double helix. uh, Most of the time, almost 99% of the time, DNA is a double helix, but when there is a transcription or when there is a replication, in that case, this double helix of this DNA has to be open to access the process, to access for replicative enzyme or transcription enzyme. Okay, in that case, DNA will open up. Okay, it will only open up. Okay, but most of the time, the DNA is double helix structure. Okay, and antiparallel means, what do I mean by antiparallel? So for, let's say, this is a one strand of DNA and this is a double strand of DNA. Okay, so one strand, this is the 5 prime end and this is 3 prime end. On the other hand, this is 3 prime end and this is 5 prime end. Okay, so this is called an antiparallel fashion, where this 5 prime end of the DNA is attached to the 3 prime end. Okay, and this is called the antiparallel fashion. So DNA is a double helix structure and it is antiparallel. It runs in antiparallel fashion. Okay, okay, so base pairing, again, as I mentioned, adenine, in case of DNA, adenine always base pairs with thymine, and guanine base pair with cytosine. Okay, in case of RNA, as I mentioned, there is an absence of thymine. Instead of thymine, it has a uracil. Okay, so adenine base pair with the uracil, whereas the guanine is still base pairing with cytosine. So the only difference between DNA and RNA is that the RNA has a uracil instead of thymine. Okay, and again, as I mentioned, this base pairing occurs via hydrogen bonds. Okay, this is very important for you to know for your exam, because they often ask you question about how this base pairing occur via what bonds. Okay, so don't confuse with phosphodiester bond with the hydrogen bond. Okay, so this nitrogenous bases, they are attached to each other via hydrogen bonds, whereas the nucleotides, they are attached via phosphodiester bonds. Okay, so very important and very high yield concept for college classes. Okay. Okay, so I was keep talking about the bases, nitrogenous bases, but how they are like, how they are structured. So on the right side of the image, as you can see here, uh, this is all the bases. This is all bases, as you can see, but they are also differentiate this bases as a purine and pyrimidines. So purine includes adenine and guanine, whereas pyrimidine includes the rest of them, which is cytosine, thymine, and uracil. Okay, so purine has only two bases, whereas pyrimidines have three bases. Okay, and one another difference between purine and pyrimidine in terms of structure is that the purines have two ring structures, whereas pyrimidines have only one ring structure. Okay, so this is very important to know that the purines have two rings, whereas pyrimidines have only one ring. And the way I remember these purines and pyrimidines are, uh, the way I remember these purines and pyrimidines is that pure as gold. So pure, which means purine, S stands for adenine, and gold stands for guanine. Okay, so pure as gold. Purines are adenine and guanine, and pyrimidines are cytosine, thymine, and uracil. Okay, and if you look at carefully, uh, there is not a significant difference between the structures of these purines and pyrimidines. Okay, so if you see a structure of thymine and a structure of uracil. Okay, so in thymine there is an additional methyl group present, whereas uracil does not have any type of methyl group. Okay, so that's the only difference present. And in case of purines, there is a presence of this NH2 group instead. In the guanine there is a presence of ketone group, and again there is an presence of this amine group, whereas in the adenine there is no such a group present. Okay, and let me quickly erase this. So this is just the structural difference. Is not a huge difference, but again this is very relevant to know for the exam, that the purines are adenine and guanine, and pyrimidines are cytosine, thymine, and uracil. Okay, and now let me mention about the hydrogen bonding that occurs between these bases. Okay, so adenine always, as you know by now, that the adenine always base pairs with thymine. Okay, but how do they base pair exactly? So they are linked to each other via a two hydrogen bonds. Okay, they are linked to each other via two hydrogen bonds, whereas guanine and cytosine, they are linked to each other via three hydrogen bonds. Okay, so now let me ask you a question. Is it easier to break a bond between adenine and thymine, or is it easier to break bond between guanine and cytosine? And if you said it's easier to break bond between adenine and thymine, that means you are correct, because they have only two hydrogen bonds. So it's easier to break two bonds than three bonds. So the cell has to consume less ATP to break down these two bonds instead of these three bonds. Okay, so very crucial for the cell to use the as less ATP as possible, because cell cannot just waste the ATP in such a process if there is an alternative things available. Okay, so it's easier to break bonds between two hydrogen bond, and it's harder to break these three bonds. Okay, and if you probably heard somewhere that the DNA is negatively charged, and that is correct, that the DNA is a negatively charged, but what gives the DNA a negative charge? Okay, and the presence of this phosphate group in DNA, as I mentioned before, it's present at the 5' end, it gives the overall negative charge to DNA. Okay, it gives overall negative charge to DNA, because if you look at the structure of phosphate, it is PO4 3 minus. Okay, so this negative charge gives overall negative charge to DNA. Okay, so DNA is overall negatively charged. Okay, let me quickly erase. Okay, so let's move on to the next one. Okay, now DNA is very, we have so many amount of DNA in our cell, and the question is how can this all DNA can be fit in the small nucleus, because we have so many amount of base pairs, we have so much amount of bases present, and how this all DNA is, how can all this DNA to be packed in the small eukaryotic nucleus? And the answer relies on the fact that this DNA is wrapped twice around the histone protein. Okay, so this is a very special protein. It is an octamer actually. It has a H2A, H2B, H3, H4, and H1, which is a linker. You don't have to know. You don't have to know this in greater detail, but just know that the histone is an octamer protein, and DNA wraps twice around this protein and forms like a bead of structure, which is known as a nucleosome. Okay, so the DNA that is wrapped around histone is known as nucleosome. Okay, but the question again is that how can this histone hold this DNA around this protein, around itself, right? How can this histone hold this DNA around itself? And the answer again relies on the fact that DNA is negatively charged due to phosphate group, as I mentioned already. But in histone, there is a positively charged amino acid such as arginine and the lysine. So they are positively charged amino acid. They're present on the outside of this histone protein, and the DNA is negatively charged, so they are attracted to each other and they form a strong bond between each other. Okay, and that holds the DNA around this histone protein. And just imagine if there is a mutation on this histone protein, such as in the lysine or arginine, whichever, right? If there is a mutation present, in that case DNA cannot be wrapped around this protein, and DNA will be in the loose form. Okay, and it will be very hard to pack this DNA within the small nucleus of the eukaryotes. Okay, so it is very important for the cell to have this positively charged amino acid, particularly in the histone protein, so that the DNA can wrap around twice in the histone protein. Okay, and again, as I said, this packaging allows the DNA to be stored in small nucleus, because DNA is very quite large, so it is very important to be packed around this protein. Okay, so the take home here is that this histone has a positively charged amino acids such as lysine and arginine, and DNA wraps twice around this protein, and the complex of histone and the DNA is known as nucleosome. Okay, so let's move on to the next one. Again, this is something that I've already mentioned. The DNA and the histone protein, they form a complex known as a nucleosome, and the DNA that is held within this histone protein is known as chromatin. Okay, so let me go back to the previous slide. As you can see here, this beads itself is known as a nucleosome, but if you look at the whole strand, this is known as the chromatin, right? So this is very important to understand that the nucleosomes are nothing but just the bead-like structure where the histone protein, where the DNA is wrapped around the histone protein. Whereas if you collect and if you add these all bits together and forms like a big strand, the open strand, it's known as chromatin. Okay, and it turns out that there are two types of chromatin, depending on the situation, like in when the cell needs a transcription, right? And we will talk more about the transcription. It's a process by which DNA is used to make the mRNA, and mRNA is used to make the proteins. Okay, so euchromatin is a structure of the DNA where it is a transcriptionally accessible, which means it's a loosely it's a, like a pretty much an open DNA where this transcription enzyme can easily access the DNA to make mRNA. Okay, whereas heterochromatins are transcriptionally inactive because they are condensed form of chromatin. So the chromatins are condensed. Since chromatin is condensed, the transcriptional enzyme cannot access the DNA, so it prevents the transcription to occur. Okay, and if you look at this heterochromatin in electron microsome, it looks like a dense, like a dark spots. Okay, and why it looks like a dark spots, because as I said, the DNA is in the condensed form, so it is hard for the transcriptional enzymes to access this chromatin. Okay, so very important to know, because there are many epigenetic changes that happens that can alter the gene expression without even changing the DNA sequence. Okay, and we will talk about in the next slide about this. So what is this epigenetic modification? So modifications are nothing but this modification allows to change the level of gene expression. uh, it allows to regulate the gene expression without even changing the DNA sequence. Okay, so it doesn't change any DNA sequence. The sequence remains the same. It only changes the expression, the gene expression. Okay, and it turns out there are two processes, methylation and acetylation. Okay, methylation will add methyl group to DNA, which actually causes the gene to be suppressed. Okay, so it silences the gene expression. And how it silences the gene expression? So methylation allows the DNA to condense. Okay, and since DNA is condensed, the transcriptional enzyme cannot access the DNA to make the mRNA. Okay, and since it cannot do that job, mRNA level will be going down, which actually reduces the gene expression. So the gene cannot make the protein because there is a sterically hindrance for this transcriptional enzyme to access the DNA. Okay, and again, if you look at it, heterochromatin structures are predominating. And if you look at intuitively, it makes sense because, as I mentioned in the previous slide, that the heterochromatins are nothing but just the condensed form of DNA. Okay, and since DNA is condensed, these enzymes cannot access DNA, so transcription will go down, which actually reduces the gene expression. Okay, so that is a one epigenetic change. And as you can see here, that we did not change the DNA sequence. All we did is just to change the packaging of the DNA, and we reduce, or we will increase here in this later. We will talk about later how we will talk later in this slide that how they are actually increase the expression. Okay, so we did not change the sequence. All we did is just change the packaging. Okay, on the other hand, there is another process, which is known as acetylation. This process adds the acetyl group to DNA. Okay, this enzyme adds actually acetyl group to DNA, which actually opens up the DNA. Okay, so it just opens up the DNA, so the transcriptional enzyme can easily access. It can make mRNA, can make protein, so it increases the gene expression. Okay, and again, if you think it predominates the euchromatin structure, because again, euchromatin structures are nothing but just the loosely form of DNA. It's just the open version of DNA, so this enzyme can easily access and make mRNA, so gene transcription can increase. Okay, so again, in summary, epigenetic modification is nothing but a change in the level of gene expression without changing the DNA sequence. Okay, so this is the take home that you need to know. And that's it for this discussion. We will talk later about the transcription and then translation process. Okay, thank you.